Hello, I'm a biology teacher, Gulnaz Uspanova. I will show you how to effectively use graphic organizer of CLIL technologies during lesson. Let's start this lesson with problematic question. Sickle cell anemia is very abundant in African countries, also in the USA, India, Turkey, and in Italy. This disease is expressed in the fact that red blood cells are not round but sickle-shaped, resulting in less oxygen being transported. Question: what causes red blood cells to sickle? Thanks to the think, pair, share method, students first discuss the topic individually, then in pairs, and then in teams. Each discussion is given 30 seconds. During a group discussion, I write down the students' opinions on the board. Among these responses, I mark those that are most relevant to the topic of the lesson, and therefore we determine the topic and the purpose of the lesson. Due to distance learning, lessons are being held in teams. Using Teams, I can show how to present presentations for the lesson. To do that, I open the class group. The presentation has to be downloaded in advance. I open that presentation. Topic: Mutation and detectives. Lesson objectives: describe the mechanism of the formation of chromosome and the genetic mutations. In order to complete the answer to this question, I want students that we will return to this question at the end of the lesson. To work with keywords of the topic, the students perform the matching task in pairs. Go to the previously created channels, click on the Meetings button, and go to a new page for negotiations. One of the students in the pair will share the screen, go to the link in the channel, and complete the task together with their pair. The teacher will go to each channel and give feedback. After working with keywords, we start investigating the problem of mutation. Research problem. Recently, cases of genetic mutations like sickle anemia, Huntington, cystic fibrosis have increased in villages. They are being investigated by ecologists, doctors, and scientists. They are called detectives. Today, we are going to be detectives for 3 groups we created. The first group is called scientific detectives. They work with a virtual lab. Task: using a virtual laboratory and information from the internet, create types of mutations of genes and chromosomes, as well as their mechanisms. Draw the collected material on the concept map. Paying attention to the descriptors, mark your steps. The second group will be doctors' detectives. Task for them: consider the causes of diseases such as sickle anemia, cystic fibrosis, Huntington's, and draw its own fishbone. The third group, environmental detectives. Task: determine the causes of genetic mutations from the environment. The tasks in each group are with descriptors. Tasks are preloaded on channels. According to them, detectives perform their work in groups for 8 minutes. The teacher goes to each channel and gives feedback. When the time runs out, students go to the general channel and make presentation on their work. Between groups, their questions answers, they are evaluated according to descriptors. Go back to the objective of the lesson and again consider the initial problem question of the lesson. Complete the answer. Using the site padlet.com, we conduct a reflection on today's lesson. Students write their opinions so you can determine the direction for the next lesson. Thank you. Goodbye.